I'm Marta del Bianco. I'm a researcher at the Italian Space Agency. And unfortunately for you, I'm a biologist. And I didn't realize that I gave that title. The working title of my presentation is much shorter. And today I'm gonna give you an overview of the use of CubeSat technology for space biology applications. So a little bit of history. Let's see if it works. Okay, so you all know that CubeSats are really small, and being really small, to be able to perform biological experiments, we need to adapt a system that is capable of maintaining the life of whatever organism we're using, while being able to detect variation in growth or metabolism or activity. Life equals liquid water, so the majority of the space biology research CubeSats were based on microfluidic systems. So the first one was GeneSat-1, that was launched back in 2006. It was a 3-unit CubeSat, and it carried a lab-on-chip, so a micro laboratory system that we used to follow the growth and metabolic function of E. coli bacteria. Okay, so this is the chip, so the microfluidic system where with the wells where the bacteria were placed, and the input and output of the microfluidic system. What did GeneSat was commissioned by NASA, and it flew on low Earth orbit. What did the science? And it had two different imaging systems. One that looked at the density of the culture to understand how many bacteria were in the well, and one that looked at the fluorescence light that these bacteria were generating by producing the green fluorescent protein. And what they saw was that despite the microgravity conditions, despite the radiation conditions, bacteria were able to grow and express the green fluorescent protein in space. So these are the flight, the flight data through time. But they didn't, at least I didn't find any control, like ground control. This next biological study based on another CubeSat called Orios-Seslo, so organism exposure to orbit stress, was based on another bacteria, Bacillus subtilis. It was still on low Earth orbit, but a slightly higher orbit. And it followed these bacteria that were grown in these 24 micro wells. You can see here so I'm going to move so the bacteria were grown in these little wells, and there was an LED light to detect the growth of these bacteria. And what did they? The bacteria were actually. Bacillus subtilis is one of those bacteria that makes spores to basically maintain life when there are harsh conditions. So the payload was prepared with B. subtilis spores, and the spores were activated at different intervals after the CubeSat arrived in orbit. This was to check if the radiation environment was affecting the capability of germinating of these spores. And what they saw was that actually the spores, which are very hardened and very resistant to radiation, were retaining the ability of germinating in space even after almost 3 months of exposure to the space environment. However, once they germinated, as you can see here, these are the ground control and these are the flight experiments. The bacteria were really, really slow. So what normally happens when you have a bacterial growth, especially if you don't change the substrate, the bacteria will grow and then they reach basically maximum capacity and then they start dying off. And what they saw was that this was not happening in space because the growth was so slow and the metabolism was so slow that the bacteria were able to keep on growing. Basically, in the time period that they were observed, they were not reaching the downward curve. So this downward part of the growth curve. So this was really interesting from a biological point of view. This slow in the metabolism and the growth, which was induced by the space environment. And this was, for example, really relevant for actually a previous CubeSat mission, which was based on yeast. So, the same type of microfluidic system. So you have a chip with, in this case, C. cerevisiae grown in microwells with a light to detect LED light and a detector to assess the growth of the yeast through time by basically seeing how much the absorbance changes, because of course the more organisms, so yeast or bacteria there are in the culture, the more opaque the culture is. And so you can see that the chip had loads of wells, and the reason why this chip had a lot of wells that was because the scientists were testing the response of this yeast to an antifungal substance. What did they find? Actually, it's really interesting. They found that, again, the growth of the of the yeast was really slowed down by the space conditions. And since the the growth and the metabolism were so slowed down, you actually had a decreased effect of the high concentration of antifungal compound. So this shows that the effects of the space exposure, yes, on one side they can slow down the metabolism and the growth of these microorganisms, but on the other side, these can have complex implications for their, for example, from a biomedical point of view. These 3 CubeSats that I described were all 3 units CubeSats. The most recent CubeSat deployed by NASA is BioSentinel. It was launched 16th of November last year, 2022, from Kennedy Space Center. It has a very interesting orbit. It's actually all of the other CubeSat were heliocentric, Earth-centric, while this one is heliocentric, and we'll have a pass-by to the moon. So it will leave the Earth orbit and do a flyby of the moon. This one is a six unit CubeSat, therefore is much heavier, and again it will carry Saccharomyces cerevisiae, so a yeast, and it will detect how much radiation affect the DNA stability of this organism. Of course, the launch was just over 7 months ago, so there are still no biological results around. So we will keep on looking at the literature to see if like the results of this experiment. So regarding what we have been doing at the Italian Space Agency, I've been at ASI since September 2019, and when I arrived, there was great enthusiasm about the biggest sea maiden flight. On board of the biggest sea maiden flight, of course, there was the LARES-2 as the main payload, but as secondary payload, there were 4 CubeSats. 2 of which were Italian CubeSats. Both of them were 3 units. The first one is Astrobio CubeSat. Actually, I have the launch happened the 13th of July last year from Kourou in French Guiana, and I have. I'm just going to show you the. I don't know if you can hear the audio, but this is basically the ESA base.
Not the audio, but we can kind of see the video, Marta. So
okay,
I see
that was the countdown and the launch. And this is our CubeSat going to space. So this was the Vega-C was successfly launched, and it didn't go to low Earth orbit, or as we described for the previous 3 units CubeSats, but it went to medium Earth orbit, so around 6,000 kilometers. And I'm going to tell you in a minute why that is interesting. I mean, you probably know. So the first CubeSat that was on the Vega-C is Astrobio CubeSat. Again, is a 3-unit CubeSat based on a lab-on-chip technology. The scientific aim of the biological payload was to demonstrate that increasingly complex bioluminescence reactions could happen in space and in medium Earth orbit. It was based on this paper origami chip, where the reagents were spotted and dried, and basically when the experiments was started, pumps would allow the water to percolate between the different layers of the origami, and they would then mix the reagents to start the reactions. The other CubeSat, which was more involved in, is DreamCube. That was a collaboration between ASI, La Sapienza, the University of Rome, the University of Naples, Federico II, and Enea, which is is a very long acronym, but it's a research institution in Italy. So why 6,000 kilometers, a very interesting orbit, especially for a biological payload? That is because, as you probably know, we are full in the Van Allen belts. So there is very high radiation environment compared to the low Earth orbit, so where the ISS is. And of course, from a technical point of view, this is challenging, especially because you have to think that both CubeSats were built with off-the-shelf technology. And but from a biological point of view, of course, this is going to be very challenging. For Green Cube in particular, we actually tried to grow a very complex system, because as you have seen so far, the type of organism that have been applied to CubeSat missions were unicellular organisms. Of course, they were eukaryotes, so there were yeast, but never multicellular complex organisms. Actually, there were some fern spores, but they were not germinating. They were just being checked. Of course, plants have been grown in space. We know that plants are grown on regularly on the ISS. We know there has been attempts to grow plants on the moon, for example, but nobody has ever tried to grow plants on a fully automated CubeSat mission. So the Green Cube CubeSat was a 3-unit, and it harbored a growth chamber, which was just below 1 liter. I think it was 0.8 liters in volume, and had a closed hydroponic system with, of course, all of the sensors to detect the oxygen, the pressure, the temperature, so the environmental condition within the growth chamber. And it was equipped with a very bad infrared camera, but a very good camera in visible light. And the aim was to grow plants. These were Lepidium sativum, so cress, watercress, plants to microgreen stage. So for those of you who have never heard of microgreens is a developmental stage of plants, which happens after the sprout, when the leaves, the embryonic leaves, are completely developed. And the reason why we chose microgreen was, first of all, because they're very fast to grow. It takes about 10 days. And because from a human exploration perspective, they're really rich in antioxidants. So they are being targeted as a very good food source for future explorations. One thing that makes us, of course, really happy. The Jupiter on Green Cube has been working from almost from day one. Well, we ask people not to use it while we're downloading the results, but is being used all over the world, from radio amateur to communicate with one another. And we're really happy about the enthusiasm that everyone is having for Green Cube. So to conclude, CubeSat technology can be a very cost-effective way of performing biological experiments in space, especially because it allows us to access conditions that would otherwise be impossible to achieve. For example, medium Earth orbit. Of course, the CubeSat technology, like the construction of the CubeSat, is available off the shelf and is affordable. But to really do interesting biological experiments, there is loads of work that is being done to develop miniaturized systems that can be implemented on board of these CubeSats. And I report here like 3 examples that are being developed in Italy. So Kaiser Italia is developing a fluorescent microscope. So you've seen that we were able to detect fluorescence in the past, but of course there was no resolution. It was just how much light can we detect. And this fluorescent microscope is gonna fly, we don't know when, on a CubeSat in the Borealis mission. And I think the developer is again the University of Rome La Sapienza, the School of Aeronautics, I'm not sure, and the PI is Mara Mirasoli from the University of Bologna. The University of Naples is developing a cytometer to basically detect the shape and the internal structure of cells, which can be applied to CubeSat mission, while the Fondazione Amaldi, the Amaldi Foundation, is developing a Raman-based spectrometer, like a miniaturized one. And of course, CubeSat technology can be used and is being used as a payload, a hardware payload for experiments on board the ISS. We have two examples, the Italian Space Agency. One is OvoSpace, which is investigating the effect of space exposure to the granulose and theca cells of the mammalian ovaries, and they are based, and the hardware is based on CubeSat technology. I think it's a 3-unit, no, it's a 2-unit CubeSat, because of course, like many systems don't have to be implemented if you fly on the ISS. Another project that I'm actually supervising is Space Lime, which is using CubeSat technology to develop as payload to grow Physarum polycephalum, which is a slime mold on board the ISS. And since our colleagues from Japan are connected, you might know Physarum because it's a very interesting microorganism. And I don't know how many of you know this, but despite being, so can you see it is this yellow thing, and is one giant cell that shows intelligence. And here you can see scientists that in Japan have used it to reproduce the Tokyo subway system, and apparently I did a better job than the Japanese colleague. So it's a very remarkable microorganism. And with this, I would like to thank you for your attention, and I'm open to any questions.